Dear students, as proteomics evolved, people started realizing the need for open data formats. Now, every instrument, every mass spectrometer that was there in the research labs was outputting data in its own unique raw file format. So, Matrix Science, a company, came up with the MGF format, or the mascot generic format. As is obvious from its name, it's a generic format, so it can incorporate data from multiple mass spectrometers, which may output files in different raw formats and represent them in a standardized way. Let's take a look at the MGF format. So as I just mentioned, the MGF is a simple, human-readable, standardized file format, and it includes a lot of attributes that can be measured from the sample in the mass spectrometer's chamber. The company Matrix Science that incorporated this format has evolved this MGF file format along with the mass spectrometer instruments evolution, and there are multiple formats of MGF or versions of MGF now available in the market. I will elaborate what MGF format is by this table. As you can see, in the first column, we have the attributes for each of the measured quantity in the mass spectrometer's chamber. The first is the accession. So the accession number is essentially the database entries to be searched. So you have to search your sample within a database, and these accession numbers are accession numbers of the proteins in the database. Secondly, the charge, the attribute. So the charge attribute is essentially the charge on the peptide of the protein that is under observation. Next, there is the CLE attribute and the COM attribute, which gives you the enzyme that is used or the search title that you can give for your search. Next, you can look at the cutout attribute, the precursor removal. So it just gives you the precursor removal mass, the amino acid composition, the database, that is the DB attribute. It gives you the database you want to search in your query. There is a decoy attribute as well. So if you want to perform a decoy search, then you can have data here as well. The error tolerance. So in this case, you can specify if your search is error tolerant and you can accept some plus or minus mass in the search. Next, the E tag or the error tolerant sequence tags. So the sequence tags that are reported by the search may contain slight deviations, which you can tolerate if you want by setting this attribute as well. Next, the format. So here, if you look closely, the format attribute gives you the MSMS data file format. Now, this is your data which is coming from the mass spectrometer in terms of the mass over charge ratio and relative abundance. This data is dependent upon the type of mass spectrometer, and therefore there are multiple formats that can be used for setting this attribute. Next, you have the frames, you have the instrument, and of course you can specify each one of them yourself. And there are some other variable modifications that you can specify, the fragment ion tolerance. So all of these parameters you can set up in your MGF file that is output from the Matrix Science mascot search engine. Moreover, you can have attributes such as mods, mass, and pep mass. So in mass, you specify the mono, isotopic mass, or the average mass, whichever you want to use in the search for the molecule. The mods include the fixed modifications, the post-translational modifications that may be there on your protein. The pep mass is the peptide mass, which you can specify if you're looking for the peptides. Lastly, you can have a precursor mass attribute also set. So this precursor mass gives you a guesstimate of the molecular weight of the peptide or protein that has been injected into the mass spectrometer's chamber. So this can be used by your search algorithm later on to come at a better conclusion. You can also look at the quantitation data by looking at the relative abundances. And then there is the raw file identifier, which tells you the raw file format. So all of this data is there in your MGF file, that is the mascot generic format file, and you can convert this MGF file into any, you can link this MGF file into with any search engine, and you can search your mass spectrometry data. However, as I mentioned in the beginning, due to the evolution of mass spectrometers, the MGF file attributes have also evolved and grown. So now there are a lot of MGF attributes, and they keep getting upgraded. So before using an MGF file, you may want to look at the version of that MGF file that has been output and process it accordingly.